Good afternoon, everyone. My name is Dr. Ritu Parnashaha. I am an assistant professor from the Department of Life Sciences, Christu Jayanti College, Bangalore. Today, I will be discussing enzymes, its nomenclature, and classification. Enzymes are biological catalysts which are protein in nature, thermolabile, and are synthesized by living cells. And the substances on which enzyme acts are known as substrates. The major similarities between inorganic catalysts and enzymes include that they speed up chemical reactions, accelerate state of equilibrium, doesn't change after the chemical reaction, lowers the activation energy, results in the formation of reaction intermediate, and are influenced by external factors. The major differences between inorganic catalysts and enzymes include inorganic catalysts are chemical in nature, which includes metals, metal oxides, inorganic compounds, whereas enzymes are always protein in nature. Inorganic catalysts are generally synthesized from chemical sources, whereas enzymes are synthesized by living cells. Inorganic catalysts can perform their catalytic activity under harsh conditions like high temperature, high pressure, whereas enzymes can only carry out their catalytic activity only under mild conditions. Inorganic catalysts have very low specificity, whereas enzymes have very high specificity. The activity of inorganic catalyst cannot be regulated, whereas enzymes activity can be regulated by covalent modification or allosteric regulation. Inorganic catalysts are much smaller in size, whereas enzymes are much larger in size. The turnover rate of inorganic catalysts are quite low, whereas the turnover rate of enzymes are much higher. And inorganic catalysts have a higher lifespan. They have a much longer shelf life, whereas enzymes can easily get denatured as well as they can lose their activity. The major characteristics of enzymes include that sometimes certain enzymes, to perform their catalytic activity, they require to bind with certain non-protein molecules. These non-protein molecules are known as cofactors. If these non-protein molecules are organic in nature, then they are known as coenzymes. So these non-protein compounds that bind via covalent or non-covalent bonds, they are also known as prosthetic groups. So any non-active enzyme or the protein part of the enzyme that is without its cofactor is known as the apoenzyme, whereas the enzyme along with its cofactor is known as holoenzyme. Now, in the early days, when enzymes were getting identified and discovered, these enzymes were classified based upon the substrate that was acted upon by the enzyme, or the type of reaction catalyzed, or the substrate that is, or the substance that is synthesized, or the chemical composition of the enzyme, or the substance that is hydrolyzed and the group that is involved. But in the recent years, it was ultimately decided upon by the International Union of Biochemistry and Molecular Biology that enzymes be classified on the type of chemical reaction that they catalyze. On that basis, the enzymes are classified on 6 groups. First is the oxidoreductase, which is basically the enzymes that perform oxidation reduction reactions. Examples include cytochrome oxidase, lactate dehydrogenase. Transferase enzymes helps in the transfer of functional groups such as amino group, acetyl group, or phosphate group. Examples include acetate kinase, transaminase. Hydrolase enzymes performs hydrolysis. Examples include lyase, sucrase. Lyase enzymes helps in the removal of groups of atoms without hydrolysis. Examples include oxalate decarboxylase, isocitrate lyase, etc. Isomerase enzymes helps in the rearrangement of atoms within a molecule. For example, alanine racemase. Whereas ligase enzymes helps in the joining of two molecules, preferably using energy that is derived from the breakdown of ATP. Examples include acetyl-CoA synthetase, DNA ligase. After this classification, each and every enzyme that is identified, they are actually given a specific EC number, or which is also known as enzyme commission numbers. For example, each enzyme gets 4 enzyme commission number, which is denoted by, as you can see on the screen, as 1.1.1.1. The first number, 1, which is blue in color in this picture, it always identifies or denotes the main class of the enzyme. Whereas the second number denotes the subclass, which actually tells us that what type of compound or bond the enzyme acts on. And the third number identifies or denotes the subclass, which is a further classification. And the last number basically is just a serial number that the enzyme has within that enzyme commission class. Some examples include like cytochrome C oxidase. It has an enzyme commission number as 1.9.3.1. 1 meaning it belongs to oxidoreductase, 9 meaning it acts on a heme group of donors, and 3 meaning that it actually acts on oxygen as an acceptor. Another example includes tripeptide aminopeptidase. The enzyme commission number is denoted as 3.4.11.4. So the 3 is denoted by hydrolase, 4 denotes that the enzyme acts on peptide bonds, whereas 11 denotes that it cleaves off the N-terminal amino acid from a polypeptide, whereas 4 is its serial number within this group. So in conclusion, we understood today that what are enzymes, what are their similarities and differences with inorganic catalysts, how enzymes are classified, and how enzyme commission numbers are denoted to different types of enzymes. Thank you.